Hi everybody. Today we are going to say about screening and strain development strategies. Introduction. The microorganism with extraordinary biochemical characters has been used for production of commercially valuable products in fermentation industries. The first step in the screening of potential microorganisms with industrial application is the isolation. Obtaining either pure culture or mixed cultures with desired characters or to produce the desired product. First, we'll see about screening method. Primary screening of microorganisms. Primary screening may be defined as detection and isolation of desired microorganisms based on its qualitative ability to produce the desired products like antibiotic or amino acid or an enzyme. The following are some of the important primary screening techniques: the crowded plate technique, indicator dye technique, enrichment culture technique, oxanotrophic technique, technique of supplementing volatile and organic substrates. First method, the crowded plate technique. This technique is primarily employed for detecting those microorganisms which are capable of producing antibiotics. Natural substrates like soil or other sources consisting of microorganisms is selected and serially diluted. Suitable aliquot of the serial dilution is chosen to produce 300 to 400 individual colonies when plated on an agar plate after incubation. Such a plate is called crowded plate. The antibiotic producing activity of a colony is indicated by no growth by the formation of a clear and colorless area around the antibiotic producing microorganism colony on the agar plate. Colony is isolated from the plate and purified either by making repeated subculturing or by streaking on a plate. The purified culture is then tested for its antibiotic spectrum. Indicator dye technique. Microorganisms capable of producing acids or amines from natural sources are detected using this method by incorporating certain pH indicator dyes such as neutral red or bromothymol blue into nutrient agar medium. The change in the color of a particular dye in the vicinity of a colony indicates the ability of that colony to produce an organic acid or base. Enrichment culture technique. This technique is generally employed to isolate those microorganisms that are very less in number in a soil sample and possess specific nutrient requirements and are important industrially. This was first designed by Bejernik to isolate the desired microorganism from a heterogeneous microbial population. The following are the steps to the enrichment culture technique. Nutrient broth is inoculated with the microbial source material and incubated. A small portion of the inoculum is plated onto the solid medium and well-isolated colonies are obtained. Suspected colonies from a plate are subcultured on fresh media and subjected for further testing. Example of the enrichment culture medium: cellulose medium for cellulase production and tributane medium for lipase producer. Oxanotrophic technique. This technique is employed for the detection and isolation of microorganisms capable of producing certain extracellular substances such as growth stimulating factors like amino acids, vitamins. First step is the preparation of first plate. A filter paper strip is put across the bottom of petri dish. The nutrient agar is prepared and poured onto the paper disc and allowed to solidify. Soil sample is diluted and proper dilutions are inoculated and incubated. Preparation of second plate. A minimal medium lacking the growth factor is prepared and seeded with the test organisms. The seeded medium is poured onto a fresh petri dish and the plate is allowed to set. The agar in the first plate is then carefully lifted with a spatula and placed onto the second plate without inverting. The growth factors produced by the colonies present on the surface of the first layer of agar can diffuse into the lower layer containing the test organism. The zone of stimulated growth of the test organism around the colony is an indication that the organism produces growth factors extracellularly. Technique of supplementing volatile organic substance. This technique is employed for the detection and isolation of microorganisms capable of utilizing carbon source from volatile substrates like hydrocarbons, low molecular weight alcohols, and similar carbon sources. Suitable dilution of a microbial source like soil suspension are spread onto the surface of sterile agar medium containing all the nutrients. The required volatile substrate is applied onto the lid of the petri dish, which are incubated by placing them in an inverted position. Enough vapors from the volatile substrate spread to the surface of agar within the closed atmosphere to provide the required specific nutrient to the microorganism, which grows and forms colonies by absorbing the supplemented nutrient. The colonies are isolated, purified, and stock cultures are made which may be utilized for further screening tests. Next, we'll see about secondary screening of microorganisms. Microorganisms isolated in the primary screening are critically evaluated in the secondary screening so that industrially important and viable potentialities can be assessed by the following methods. To determine the product produced by an organism is a new compound or not, a determination should be made about the yield potentialities of various isolated microorganisms that are detected in primary screening for that new compound. It should determine about the various requirements of the microorganism, such as pH, aeration, temperature. It should detect whether the isolated organism is genetically stable or not. It should reveal whether the isolated organism is able to destroy or alter chemically their own fermentative product by producing adaptive enzyme if they accumulate in higher quantities. It should reveal the suitability of the medium or its constituent chemical formula for the growth of the microorganism and its yield potentialities. It should determine the chemical stability of the product. It should reveal the physical properties of the product. It should determine whether the product produced by microorganism in a fermentative process is toxic or not. Secondary screening should reveal that whether the product produced in fermentation process exists in more than one chemical form. Chemical formation of additional products is important since their recovery and sale as byproducts can greatly improve the economic status of the fermentation industry. The new organism should be identified to the species level. This will help in making a comparison of growth pattern, yield potentialities, and other requirements of the test organism with those already described in the scientific and patent literature as being able to synthesize products of commercial value. It should select industrially important microorganisms and discard others which are not useful for fermentation industry. It should determine the economic status of a fermentation process undertaken by employing newly isolated microorganisms. First method in secondary screening is giant colony technique. This technique is used for isolation and detection of those antibiotics which diffuse through solid medium. Species of Streptomyces is capable of producing antibiotics during primary screening are isolated and inoculated into the central area of a sterilized petri dish containing nutrient agar. The plates are incubated until sufficient microbial growth take place. Cultures of the test organism whose antibiotic sensitivity is to be measured are streaked from the edges of a plate and further incubated to allow the growth of the test organisms. The growth of different test organisms is inhibited by the antibiotic secreted streptomyces is measured in millimeters. Next method is the filtration method. This method is employed for testing those antibiotics which are poorly soluble in water or do not diffuse through the solid medium. The broth containing streptomyces and its mycelium is separated by filtration to get culture filtrate. Various dilutions of antibiotic filtrates are prepared and added to the molten agar plating medium and allowed to solidify. Then cultures of various test organisms are streaked on parallel lines on the solidified medium and incubated. The inhibitory effect of antibiotic against the test organism is measured by their degree of growth in different antibiotic dilutions. Next method is the liquid medium method. This method is generally employed for further screening to determine the exact amount of antibiotic produced by an microorganism like streptomyces. Erlenmeyer conical flasks containing highly nutritive medium are inoculated with streptomyces and incubated at room temperature. They are also aerated by shaking continuously and vigorously during incubation period to allow streptomyces to produce the antibiotic in an optimum quantity. Unstable, but it should be genetically stable. It should be easy to perform genetic manipulation on it. Unfortunately, the organism isolated from the environment for industrial use does not fulfill all these requirements. Any property whose addition or deletion from a strain results in minimizing its cost. Production comes under the purview of strain improvement. It can be increased in byproduct formation, release of intracellular byproduct by altering permeability of cells, overcoming feedback inhibition, repression phenomenon, simplifying procedure for extraction, purification of bioproduct from fermentation medium. Hope you have gained knowledge regarding this screening and strain development strategy. Thank you all for your patience listening.